Hi. So now that we've spent some time talking about protein structure, we are now going to discuss the globular proteins myoglobin and hemoglobin as an example of how protein structure affects function. So as you may recall from physiology, hemoglobin and myoglobin are both responsible for the transport of oxygen throughout the body. In this unit, we'll explore how both the tertiary and quaternary structure of these proteins affects their affinity for oxygen and how they work together to help deliver oxygen to our muscles and other various tissues. In class this week, we'll actually spend 2 meetings working on section 8 from your workbook because it's sort of a long section, so we're going to break it up in part 1 and part 2. Again, this is section 8 entitled Hemoglobin, Protein Structure and Function. So through these activities, we will be able to analyze and describe hemoglobin's oxygen binding characteristics using a graph and then correlating the graph with the protein's function. We'll also define and describe the terms cooperative binding and P50 value. We will then describe how conformational changes of hemoglobin caused by the allosteric binding of oxygen is important to hemoglobin's function, and then apply our knowledge about protein structure and function relationships to predict how changes in hemoglobin's primary sequence might lead to pathological abnormalities in hemoglobin function. So let's go ahead and get started with the video.
Here are our learning objectives for this chapter. Be sure to read over these objectives before and after the lecture, and then after our activity and reading the material, make sure that you can meet these various learning objectives as you prepare for the exam. Hemoglobin and myoglobin both bind and transport oxygen through the body. Hemoglobin transports oxygen through the blood, while myoglobin facilitates the diffusion of oxygen from the blood into muscle cells. Both proteins utilize a heme prosthetic group in order to help transfer oxygen. Where myoglobin is a monomer of 153 amino acids, hemoglobin is a tetramer of 4 myoglobin-like monomers. More specifically, hemoglobin is a dimer of dimers, where 2 alpha-beta pairs come together to form the tetramer. Again, as you can see, myoglobin is comprised of 153 amino acids, contains 8 alpha helices, and a heme prosthetic group. The heme prosthetic group is a porphyrin ring system that coordinates an iron atom in its center. Let's take a closer look at the heme prosthetic group, shall we? In the center of this conjugated ring system of the porphyrin, the central iron atom is coordinated within the plane by the 4 nitrogens of the pyrrole groups. The remaining 2 coordination sites above and below the iron atom are occupied by oxygen from above and histidine F8 from below. The oxygen molecule is further stabilized by hydrogen bonding with histidine E7 and then locked into place with valine E11 and phenylalanine CD1. Valine and phenylalanine can actually swing in and out as hemoglobin breathes to facilitate oxygen movement on and off the heme. When oxygen is not present, a water molecule takes its place. In free heme, or heme not associated with the protein myoglobin, the iron 2 atom in the heme is irreversibly oxidized to iron 3 after exposure to oxygen. Once oxidized to iron 3, oxygen can no longer bind. Therefore, the presence of myoglobin prevents this oxidation of iron 2 to iron 3 in the presence of oxygen. Otherwise, myoglobin or hemoglobin would not be very effective oxygen transport molecules. In addition to oxygen, other molecules such as carbon monoxide and nitrous oxide can bind to the heme group. Free heme, or heme not associated with myoglobin, has 25,000 times higher affinity for carbon monoxide than for oxygen. When the heme is associated with myoglobin, this affinity for carbon monoxide is reduced, but it is still 250 times greater than its affinity for oxygen. The reduction from 25,000 to 250 times higher affinity is because of the steric effects introduced by histidine E7. Histidine E7 causes the linear molecules, such as oxygen and carbon monoxide, to bend at a 120-degree angle in respect to the porphyrin ring system instead of binding perpendicular. While oxygen can tolerate this bend a little better than carbon monoxide, we can still see a greater binding affinity of carbon monoxide to both myoglobin and hemoglobin than oxygen, which makes carbon monoxide poisoning so lethal, even at very low concentrations. The binding of oxygen to myoglobin alters the overall tertiary structure of the protein. When oxygen is not present, the central iron atom is drawn out of the plane of the heme through interactions with proximal F8 histidine by approximately 0.05 nanometers. When oxygen binds, that iron atom is drawn back into the plane of the heme, which then pulls histidine F8 with it as well, which then also pulls helix 8, and eventually causes a downstream global shift in the myoglobin structure. While this structural shift in myoglobin is small, we will see a much more profound effect in hemoglobin in both global structure and binding activity after binding oxygen. The binding of oxygen to myoglobin at increasing oxygen pressure or increasing oxygen concentration can be described as hyperbolic, meaning that the binding of one oxygen molecule to one myoglobin does not affect the binding of additional oxygen molecules. Now, this makes sense because there is only one binding site for oxygen on each myoglobin molecule. But let's take the example of hemoglobin, which has 4 binding sites for oxygen. If hemoglobin showed a similar hyperbolic curve, then we would say that the binding of oxygen to the first binding site does not affect the binding of oxygen on the second, third, or fourth site. We will see, however, that this is not the case for hemoglobin, and instead of a hyperbolic curve, hemoglobin has an S-shaped or sigmoidal curve, which we will address later on. But at looking at our curve for myoglobin, we find that the P50, or the pressure at which we have 50% saturation of myoglobin with oxygen, is very low, at 2.8 torr. This means at either arterial blood pressure, which is 100 torr, or venous blood pressure, which is 30 torr, myoglobin should be at least 90% saturated with oxygen, meaning that myoglobin is able to efficiently relay oxygen from the capillaries, high pressure, high oxygen concentration, to muscle cells, which have low pressure or low oxygen concentration. Now, let's compare myoglobin to hemoglobin. Hemoglobin is a tetramer of 4 myoglobin-like subunits. More specifically, we have 2 alphas and 2 betas. One of each of the subunits comes together to form an alpha-beta dimer before complexing to become the tetramer, effectively making hemoglobin a dimer of dimers. The interaction between each alpha-beta dimer consists of 54 intermolecular interactions that stabilize the overall quaternary structure. When oxygen binds to hemoglobin, we see the same heme localized structural changes as when oxygen binds to myoglobin, where the proximal histidine is pulled with the iron atom as it is pulled back into the plane of the heme. However, this localized change causes a much more dramatic downstream shift in the quaternary structure of hemoglobin, where one alpha-beta dimer actually rotates 15 degrees with respect to the other dimer, breaking some of those intermolecular interactions that were holding the non-oxygenated hemoglobin quaternary structure. We will see how this global structural change has a crucial effect on the binding of additional oxygens. The deoxy form of hemoglobin is often referred to as the T state by biochemists, while the oxygenated form of hemoglobin is referred to as the R state. Unlike myoglobin's hyperbolic curve, the binding of oxygen to hemoglobin at increasing oxygen concentrations shows a sigmoidal relationship. These curves indicate a cooperative binding, in which the binding of one oxygen molecule does have an effect on the binding of more oxygens. In this case, we can see that it has a positive effect by increasing the relative affinity for hemoglobin to bind more oxygen molecules after the first is bound. As we can see from these graphs, the P50 of deoxy hemoglobin is 26 torr, while the P50 for hemoglobin to bind a fourth oxygen, while it has 3 oxygens already bound, is only 0.2 torr, meaning the hemoglobin has a 100-fold greater affinity for binding its fourth oxygen than it did for binding the first. Another way to look at the cooperativity of ligands binding to macromolecules is to construct a Hill plot, as we see in the lower second graph, where the ratios of the fractions bound to unbound are plotted against increasing ligand concentration. If the slope stays the same, or we have a linear line, we can see that there is no cooperativity, as we see with myoglobin. If the slope changes, as we see in hemoglobin, then we can see that there is cooperativity. If the slope increases, it is a positive relationship, meaning affinity is increased. If the slope decreases, then there is a negative relationship, or the affinity for additional ligands is decreased. The construction of the Hill plot allows us to calculate more accurate P50s for the various bound states, as we can see in the second graph. Now, you may be asking yourself, how does the binding of one oxygen affect the binding of additional oxygens at sites 25 to 37 angstroms apart? It seems like a great distance atomically to have such dramatic effects on affinity. As you recall from earlier, the binding of the first oxygen molecule is enough to shift the entire molecule from the T state to the R state. By binding that first oxygen molecule, we pull the iron back into the heme ring, which pulls histidine on helix 8, which pulls helix 8, while that pulls the entire alpha-beta dimer and drives the 15-degree rotation of one dimer against the other. This global shift to the R state literally opens up the other 3 heme binding sites and makes it easier for more oxygen molecules to bind. The reason that it is harder to bind the first molecule is because to drive that dimer-dimer rotation, we need to first break a number of intermolecular interactions that stabilize the T state. But once those non-covalent interactions are broken, it is easier to shift to the R state and then bind additional oxygen molecules. Some of these intermolecular interactions that are disturbed are further described in figures 9 and 10 of chapter 7 in your text. And we will also look at some of these interactions more closely in our activity in class. In addition to changes in oxygen concentration, the local pH and carbon dioxide concentrations of the local environment can also affect oxygen uptake and release from hemoglobin and myoglobin. As hemoglobin transitions from the T to R state, the pKa of some amino acids are decreased due to disruptions of ionic bonds during the alpha-beta dimerization twist. At physiological pH around 7.3, hemoglobin releases approximately 0.6 hydrogens per oxygen bound, or on average about 2.5 hydrogens released for a fully saturated hemoglobin. increasing the pH or decreasing the relative hydronium concentration, therefore, can cause hemoglobin to bind even more oxygen at low oxygen concentrations. This also means in capillaries where the concentration of oxygen is low and hydronium concentrations are high because of exerting muscles, excess protons can be taken up by hemoglobin in exchange for oxygen, which then can be transferred to myoglobin across the capillary wall. You will see later on in the term a number of metabolic processes such as glycolysis, the citric acid cycle, and the electron transport chain all release protons that must be buffered or transported out of the body in order to maintain physiological pH. Just last week, we saw how the bicarbonate blood buffering system can help maintain that pH, but now we can see how hemoglobin can facilitate some of the proton transfer as well. Let's look at this process step by step, starting at the lungs. In the lungs, we have a very high oxygen concentration such that we saturate nearly all of the hemoglobin in the capillaries of the lungs. The oxygenated hemoglobin then travels through the veins toward the capillaries at the muscle, where the concentration of oxygen decreases from 100 torr in the lungs to 20 torr near the capillaries. Now remember, at this point, the binding affinity for fully saturated hemoglobin is very high, as much as 0.28 torr. So to help facilitate the transfer of oxygen from hemoglobin to myoglobin, which has an affinity closer to 2 torr, the excess protons can bind oxygenated hemoglobin, driving off oxygen and stabilizing the deoxygenated hemoglobin, or T state. As we drive off oxygen and convert back to this T state, the affinity of hemoglobin to bind oxygen greatly decreases, such that the oxygen bound to myoglobin will not cross back over, but instead can be transferred on to muscle cells. Deoxyhemoglobin can then travel back through the veins, picking up protons and even carbon dioxide, releasing some as it travels through the blood to help the bicarbonate blood buffer or trigger release of CO2 and H2O through the lungs using diffusion. Or it can release CO2 and protons through the lungs directly as it picks up fresh oxygen in the lungs. Another small molecule that can affect oxygen binding to hemoglobin is BPG, or bis-phosphoglycerate. BPG also stabilizes the T state of hemoglobin, and it does so by binding to the 2 beta subunits and cross-linking them, utilizing 2 histidine molecules. The presence of BPG therefore lowers hemoglobin's affinity for oxygen. BPG is vital in allowing oxygen transfer from hemoglobin to myoglobin and is also important in mammals that live at high altitudes. At high altitudes, the decreased atmospheric pressure causes hemoglobin to take up less oxygen in the lungs, which would then cause it to unload less as it travels to the muscle. An increase in BPG allows hemoglobin to release more of that captured oxygen to myoglobin than it would without the increased BPG. This is also an important aspect in pregnancy. In mammals, fetal hemoglobin has mutated beta subunits that has a decreased affinity for BPG, allowing maternal hemoglobin to pass oxygen to the fetal hemoglobin even in the presence of similar BPG concentrations. In the fetal hemoglobin, one of the BPG stabilizing histidine residues is mutated to an uncharged serine, therefore decreasing its affinity to bind BPG. In the previous slide, we saw how a mutation in hemoglobin can be beneficial by allowing the transfer of oxygen from mother to fetus. However, not all mutations are beneficial. For example, in sickle cell anemia, a single amino acid mutation where charged glutamic acid residue 6 is mutated to a hydrophobic valine in the beta subunits, creating a sticky hydrophobic surface. This causes the deoxyhemoglobin to aggregate together into linear strands. These aggregations then cause the sickle shape of erythrocytes in the blood. If enough of these aggregates occur, painful and scary blood flow blockages can occur, causing a sickle cell crisis in patients, which can then lead to organ failure and possibly death. To read more about this mutation and how it can actually help protect people against malaria, check out this section 1E of your textbook in Chapter 7. In our in-class activity, we will revisit some of these various mutations. We'll also revisit how pH and BPG affect oxygen binding to hemoglobin and how hemoglobin facilitates CO2 respiration. See you then.